Hello students, today we are going to discuss Unit 12 Fish Diseases taken from BSc second year. In this chapter we are going to discuss about the diseases causing organisms, the methods and prevention for control of diseases, and also we learn about the health management. Fish are prone to hundreds of parasitic and non-parasitic diseases, especially when grown under controlled conditions. Adverse hydrological conditions often precede parasitic attacks, as the resistance of fish is thereby lowered. Mechanical injuries sustained by a fish when handled carelessly during fishing and transport may also facilitate parasitic infection. The prevalence of fish diseases is very much dependent on the intensity of stocking. So, when a farmer decides to raise the stock rate, he not only has to provide extra food but also has to take special care to prevent and cure outbreaks of diseases. Diseases are more common in freshwater environments as it has been found that susceptible freshwater fish are significantly free from disease when grown in slightly brackish water. Properly managed ponds usually remain free from diseases. Carelessness in stocking and feeding may result in serious parasitism and mortality. Prevention is better than cure. Care should be taken to prevent parasites gaining access to the culture ponds from any nearby infected sources. Now, if you see the few fish diseases, what would be the preliminary observations? Fishes are poikilotherms, hence, the environmental impact is more in fishes when compared to warm blooded animals. So, diseases can be diagnosed only in freshly killed fishes and live fishes. If it is late after the death of fish, diagnosis is very difficult due to the chemical changes in the body at normal temperatures. Slime production is more in diseased fishes. After death, it settles on the bottom of the pond, then floats on surface of water due to the gases produced by chemical changes in the body. Mucus samples should be collected from body surface and gills and examined under microscope. Change of body coloration, abnormal behavior of the fish, then examine the external features and later internal examination. Examine the size, color, and shape of the internal organs like liver, kidney, and spleen. Examine the fluid accumulation, hemorrhages, and inflammations in the body cavity of fish. Take out the samples from vital organs and go for bacteriology, virology, and histological studies. Then examine for tumors or swelling in the body. If you look at the viral diseases, viruses are transmitted from one host to the other through a structure called virion. Viruses are classified mainly based on external structure, shape, size, capsid structure, RNA, and DNA nucleic acids. Viruses cause disease by weakening the host tissue or by forming tumors in the host tissues. There is no treatment for viral diseases, only prophylactic measures have to be taken. Now, lymphocystis. Woodcock identified this disease in fishes, marine, freshwater, and aquarium fishes susceptible to these diseases. Tumor formation is the important character of this viral disease. Riles external lesions raised and made up of granular nodular tissue, which is composed of many greatly enlarged host cells. Mature lesions may become slightly hemorrhagic. Within 6 to 15 days of infection, the tumors grow to 1,000 times. It causes a lot of damage in the Baltic Sea area in America. Viral hemorrhagic septicemia. This disease is caused by an unequal shaped fish virus with RNA. This disease occurs in salmon fishes. Transmission occurs through the water by a flagellate. This disease is also called as infectious kidney swelling and liver degeneration In German and pernicious anemia, infectious or enterohepatic renal syndrome in France. The symptoms are kidney swelling, reduced appetite, obvious distress, erratic spiral swimming, multiple hemorrhages in skeletal muscles, change in body color, reddish fins, etc. The only control measure is prevention. Infectious pancreatic necrosis, that is HPN. This disease is found in trots. This disease causes high mortality of fish, fingerlings, and occasionally larger fish. The symptoms are darkening distension and at times hemorrhages in ventral areas, including base of fin. There is pronounced pancreatic necrosis. Chlorine at 200 ppm is effective for treatment. The third one is infective hemopoietic necrosis, IHN. This was observed for the first time in trots in British Columbia. This disease occurs more in fry and fingerlings and occasionally in adults. The symptoms are pale gills, reddish fins, black coloration of the body, abdomen swelling, and huge mortality. The symptoms are clear in 12 to 45 days after the entry of virus into the host body. Chinook disease. A small site virus is responsible for this disease in Chinook salmon fingerlings. The symptoms are exophthalmos, distended abdomen, dull red areas on the dorsal surface anterior to dorsal fin. The liver, spleen, kidney, gills, and heart are pale. The disease is transmitted by the egg from the carrier female, and there is no treatment for this. Channel catfish virus disease. This disease occurs in fingerling of catfish. The symptoms are that the fish show abnormal swimming and rotating, hemorrhagic areas on fins and abdomen, fluid accumulation in abdomen, and pale gills. There is no treatment for this disease. Destruction of infected fish may prevent spread of the disease. Next, coming to bacterial diseases in fishes. Furunculosis, vibriosis, dropsy, and fin rot. We are going to see one by one now. Furunculosis, it is caused by Aeromonas salmonicida in salmon fishes. It is a non-motile gram-negative bacterium. It frequently appears to infect a large amount of decaying matter. It is also observed in few other fishes. The first symptoms of this disease is appearance of boil-like lesions. Other symptoms are bloodshot fins, blood discharge from the vent, hemorrhages in muscles and other tissues, and necrosis of the kidney. Bursting of boils allow the spread of this disease among other fishes and also offer suitable areas for fungus growth. In acute forms, it is systemic bacterial infection, a septicemia with bacteria present in the blood, all tissues and lesions. Fishes severely infected with the bacteria die in good number. Treatment. Remove the severely infected fishes from the pond and supply food containing antibiotics like sulfonamides or nitrofurans. Sulfonamides like sulfadiazine, sulfaguanidine are given orally with food at the rate of 22 grams per 100 kgs. Other antibiotics like chloromycin, mycetin, and tetramycin are most effective at a dose of 5 to 7.5 grams per 100 kg of fish per day. Disinfect the eggs with 0.015% solution of methylate or 0.185% of acrylflavin. Next, columnar disease. It is caused by Chondrocus columnaris, and Cytophaga columnaris. In many freshwater aquarium fish, it is a long, thin, flexible gram-negative slime bacterium. This disease is often associated with low oxygen level. Initially, it is marked by appearance of grayish white or yellowish white patches on the body. The skin lesions change to ulcerations and fins may become frayed. Gill filaments are destroyed and eventually lead to death of the fish. Treatment: addition of 1 ppm copper sulfate in the pond to control this disease is effective. Tetramycin administered orally with food at a rate of 3 grams per 100 pounds of fish per day or 10 days is very effective. Deep treatment is in malachite green for 10 to 30 days seconds in 1 hour bath in 1 ppm furanase is very effective to control this disease. Fin or tail rot. Tail or fin rot disease is caused by Aeromonas salmonicid, and however protozoans and fungi may also be involved. It is characterized by appearance of white lines along the margins of fins. The capacity usually progresses towards the base, eroding them and causing hemorrhage. The fin rays become brittle fast and later break, leading to the complete destruction of the fins. The infection may also spread on the body surface. Fin and tail rot are associated with poor sanitary conditions in fish ponds and with water pollution in nature. Treatment: the fin or tail rot may be checked at an early stage by keeping fishes in 0.5% copper sulfate solution for 2 minutes. Control may be achieved with 10 to 50 ppm tetramycin and 1 to 2 ppm of benzalkonium chloride. In severe infections, the affected parts are surgically removed and the fishes are then kept in 0.04% of potassium dichromate. Vibriosis. Vibrio bacteria are the causative agents of vibriosis disease in salmon and many other fishes. This disease may occur in waters with low oxygen. These bacteria are small gram-negative bacilli, characteristically curved. Diseased fishes show large, bright colored bloody lesions in the skin and muscles, hemorrhages in eyes. Gills may bleed with slight pressure, and inflammation of the intestinal tract. Treatment: sulfamethoxine at a rate of 2 grams per 100 pounds of fish per day gives good results. Terramycin at 3 to 4 grams per 100 pounds of fish per day for 10 days also gives satisfactory results. Dropsy. Pseudomonas punctata is the causative agent of this disease. It is characterized by accumulation of yellow colored fluid inside the body cavity, protruding scales, and pronounced exothalamic conditions. This is known as intestinal dropsy. In case of ulcerative dropsy, ulcers appear on the skin. Deformation of backbone takes place and show abnormal jumping. This is a fatal disease in culture systems. Treatment: removal and destruction of fishes, followed by draining, drying, and disinfecting the pond with lime, are preventive measures to control the disease. The infected fish may be cured with 5 ppm potassium permanganate for 2 minutes dip bath. Streptomycin and oxytetracycline give good results. Cotton mouth disease. The filamentous bacteria, flexibacteria, is the causative agent of this disease. The main symptom is appearance of fungus like tuft around the mouth. This can be treated with antibiotics like 10 ppm chloramphenicol for 2 to 5 days and 0.3 ppm furanase for long-term bath. Next is tuberculosis. Mycobacterium is a disease causing agent which is difficult to diagnose without pathological examinations. The symptoms are ulcers on body, nodules in internal organs, fin or tail rot, loss of appetite and loss of weight of fish. Treatment: this can be cured with dip treatment in 1 is to 2000 copper sulfate for 1 minute for 3 to 4 days. Antibiotics are not successful. The fishes should be destroyed and potassium permanganate or lime used in the pond. Bacterial gill disease. This disease is caused by myxobacteria in salmon fish. Many bacteria are found in swollen gill lamellae, which show proliferation of the epithelium, and symptoms are lack of appetite. This disease is transmitted through water from infected fish. It can be treated with 1 to 2 ppm of Timson or 1 ppm copper sulfate. Saprolegnosis. The causative agents are Saprolegnia species and Aphanomyces species. Species affected: freshwater fish like carps and goldfish. Symptoms: formation of white cottony growth on fish eggs or tissues. Fish becomes lethargic, tires easily, and responds slowly to stimuli. Histopathologically rapid destruction of epidermis with slight inflammation are observed. This can be treated with 1 to 3 ppm malachite green for 1 hour or 1 to 500 formalin for 15 minutes. Branchion, branchion, this is also called as gill rot. This disease is caused by Branchiomyces demigrans and Branchiomyces sanguinis. It is reported to be common on cultivated fishes in ponds having abundant decaying organic matter. The tubules of fungus grow into the respiratory epithelium of the gills, causing inflammation and damage to their blood vessels. The blood supply is stopped to the infected area, as a result of which it becomes necrotic. It can be controlled with 5% common salt for 5 to 10 minutes. Ichthyophanosis. It is also known as reeling disease. It is characterized by swinging movement on the infected fish. It is caused by Ichthyophonus hofferi. It enters into the host along with the food. The spores spread to the various organs and in several cases spread out to the skin, which may rupture and become ulcerative at several places. It is extremely difficult to control this disease. The infected fishes are isolated from the stock and kept for treatment in separate ponds. Medicines like sulfamethanis, tetramycin, and calomel are useful to treat the infected fish. Next, protozoan diseases. Whirling disease. This disease is caused by a Myxosporidion protozoan, Myxosoma cerebralis, only in solvent fishes. The symptoms are pancreatic necrosis, lesions, and disintegration of the cartilaginous skeletal support of the organ of equilibrium. Rapid tail chasing type of whirling is often seen when the fish is frightened or trying to feed. The typical symptoms usually appear at 12 months after exposure to the disease. If the pond contains all infected fish, it is better to destroy them by deep burial. Then, the pond should be cleaned thoroughly and disinfected with calcium cyanamide, quicklime, or sodium hypochlorite. Costiasis. This is caused by a mastigophore, Costia necatrix, in culture fishes. This is a common disease in ponds where fishes live densely in water with a low pH and poor conditioned food. The parasites live in large numbers on fish skin, fins, and gills. The symptoms are appearance of gray, blue film on the skin, which turns to red patches in severely affected cases. The infected fish becomes weak, loss of appetite occurs, and finally dies. They can be treated with 3% common salt for 10 minutes or 1 is to 2500 formal in solution. Ichthyotriasis. This disease is caused by ciliate Ichthyothrius multifilus. This disease is also called as IH or white spot disease. The young parasites moving in water get attached to the skin of the fish. They grow between the epidermis and dermis and, after becoming large in size, fall to the bottom of the pond. Infected fish develop small white spots on the skin and the fins. These parasites attack the gills also. Fish respond by jumping in the water and rubbing their body against water objects. Respiration gets affected and they finally die. Treatment. Dip treatment in 1.5 ppm of malachite green or in 10 ppm of acriflavin gives good results. 3% salt solution, 1 is to 4000 formalin, 1 is to 1 lakh quinine hydrochloride, 1 is to 5 lakh methyl blue are also useful to treat the fish. Helminth diseases. Dactylogyrosis. The monogenic trematode Dactylogyrus is reported to cause serious infection in fishes. These are found in carps. These are found on the body, fins, and gills. The parasites start appearing in the ponds during the rains, but their prolific multiplication takes place during winter, when the intensity of infection on carp fry may reach as high as 94%. The most infected size group is 61 to 100 mm, irrespective of species. Infected fishes rest near the surface of the pond margin, swim very slowly, feel suffocation, are more slimy, dropping, and folding of fins and pale gills also we can see. Alternative baths with 1 is to 2000 acetic acid and 2% sodium chloride are effective. 10 ppm of potassium bath for 1 to 2 hours and 5 ppm in the pond may give good results. Bromex and Dilox are effective to control the disease. Gyrodactylosis. Another monotrim trematode. Gyrodactylus also causes disease in culture ponds. This also lives on fins and on the body of the fish. The symptoms are production of more slime, damage of fins, and fading of the body color. The medicines used in control of dactylogyrosis are also effective to control this disease. Other helminthes like Daphyllobothrium, Bothriocephalus, Diplostomum, Clinostomum, and spring-headed worms cause diseases in fishes. Nematodes also cause diseases in fishes, of which some of the common nematodes are Philometra and Camellanus. Leeches diseases. Leeches belonging to the Gnathobdella and Rhynchobdella attack the fishes. Leeches like Physiocola, Myzobdillu, and Hemiclepsis hold the skin of the fish and suck fish blood. After the blood meal, they detach themselves, leaving the wound open for secondary fungal infections. The growth of fish is affected and they become weak. A popular control method is dip treatment in 2.5 sodium chloride for 30 minutes. This helps to detach the parasite from the body of the host. Use of 1 ppm Dilox for 5 days. Remove the infected fishes from the pond for treatment and drain and disinfect the pond with lime to destroy the eggs and adult leeches. Then coming to crustacean diseases. Argilosis. Argillus or fish lice is a common copod parasite in fishes. It is a large ectoparasite and can move over the body surface of the fish. Argulas puncture the skin and inject cytolytic toxin through the oral sting to feed on the blood. The feeding site becomes a wound and hemorrhagic, providing ready access to secondary infection of other parasites, bacteria, virus, and fungi. Argulus transmits dropsy in fishes. In advanced stages, fish swim erratically, show growth loss, and loss of equilibrium. To control argulus, removal of submerged vegetation, wooden lattices placed in the pond, which serves as artificial substrate to deposit its eggs, can be removed at intervals to kill the eggs. 500 ppm of ammonium chloride, 510 ppm of balsam, 10 ppm of DDT for 25 seconds dip, 0.25 ppm of Dilox and 2000 ppm of Lysol for 15-second dip are effective to kill Argulus. Lerniasis. It is caused by a copod parasite Lernia or anchorworm. This disease is mostly caused by Lernia cyprinaceae. The larval stages are temporary parasites that feed on mucus and blood of fish. The adult female is a specialized fish parasite, worm-like, which burrows into the fish flesh, keeping the keeping its eggs cases protruding out of the fish body. Male Lernia do not attack the fish and are not specialized for parasitic life. Early infected fish swim erratically, flashing against the sides and bottom of ponds. Heavily infected fish swim upside down or hang vertically in the water. Only partial control of Lernia is possible with chemicals because the head is buried in the fish tissues and there are no exposed respiratory organs. Hence, prevention is more effective than control. 1% common salt eliminates larvae in 3 days. 250 ppm formalin for 30 to 60 minutes. 0.2 ppm gamaxane for 72 hours. 2 ppm of Lexone. 0.1 ppm lindane for 72 hours and 1 ppm chlorine for 3 days may give good results. Ergacillus and Salmonicola. These two parasites are responsible for huge mortality of fishes in the culture systems. These two parasites are found attached to the gill filaments and feed on blood and epithelium. Later, they may also be found on the fins and body. The infection results in impaired respiration, epithelial hypertrophy, anemia, retarded growth, etc. The fish becomes susceptible to secondary infection, especially fungus. This is a common parasite attached to the gill racers of fishes, but doesn't damage gill filaments. It can also be controlled by the above chemicals. Coming to algal diseases, Cyanophysia member Oscillatoria is responsible for fish mortality. It is found on fish body in large numbers and produce toxic substances which are responsible for fish kill. Chlorella and Pharmadium also cause discomfort in fishes. Epizoic ulcerative syndrome. It causes severe damage to India's aquaculture, especially at the moment when the Indian fisheries industry is poised for a great leap forward with high input based high tech production systems. Widespread outbreaks of disease occur suddenly and often cause mass mortality in freshwater and brackish water. Fishes causing anxiety and tremendous concern. Then environmental diseases. Environmental diseases belong to non-parasitic diseases. The environment in which the fish live and grow plays an important role for fish health. Any deterioration in the environmental qualities often creates stress to fish and favor multiplication of pathogens. Though the fish has defensive mechanism against pathogens in the form of scales, epithelial cells, acid and alkali media of alimentary canal, which offers resistance to pathogens, and filially the defense mechanisms regulated by immune system and phage cells. The pathogens predominate and diseases occur in fish farming systems. Stress response from the environment leads to fish mortality in extreme cases. At lethal level, there may be several other responses like changes in fish behavior, reduced growth, food conversion efficiency, reduced reproductive potential, reduced tolerance to disease, and reduced ability to tolerate further stress. The environmental diseases diagnosed are depletion of oxygen, the mouth remains open, gills look pale with wide operculum, bigger fishes die first. Excess carbon dioxide, excessive secretion of mucus, or high pH level in pond by epithelial cells. Excess of hydrogen sulfide gas. Pond smells like rotten eggs. The bottoms dwelling fish come up to the surface and die. Organic pollution. Dropping of pectoral fins in case of organophosphorus pesticide. Oozing of blood from eyes in some cases. All algal toxicosis. Algal bloom may appear in ponds due to accumulation of plenty of organic matter or due to excessive chemical fertilizers. Toxins released by blue-green algae like Microcystis, Anabina, and Alphanisomena kill other phytoplankton and cause surfacing of fish stock. Persistence of the bloom will cause toxicosis for the fish stock, showing symptoms like then eutrophication. Water body looks like pea soup, green in color due to the bloom of blue-green algae. Nutritional diseases. Nutritional diseases. Nutritional disorders are caused by an excess of nutrient or nutritional deficiency in fish that is more than the normal requirements. Lipids, carbohydrates, proteins, vitamins, and mineral salts are only a few of the essential nutrients for fish growth. Reduced fertility, sluggish growth, poor appetite, increased disease susceptibility, morbidity with clinical symptoms, and pathological abnormalities are some of the nutrient deficiency diseases. These are the questions that are frequently asked in the exams. Hope you will have understood. Thank you.